All right, in this video, we're going to talk a little bit about how cells get things in and out of the cell, or cell transport. So there's two types, active and passive. We're going to talk first about passive transport. So both active and passive transport are important for homeostasis, but passive transport more so. A state of relatively constant internal and external physical and chemical conditions. So it keeps the conditions inside the cell, inside the living thing, where they need to be in order for the cell and for the organism to be healthy. So one form of passive transport is diffusion, a process by which particles move from an area of higher concentration to an area of lower concentration, or we say it moves with the concentration gradient. And things move because of random motion, but there's going to be a net movement from high concentration to low concentration. If you open a bottle of perfume or put a drop of water in a corner of a container of water, you can see diffusion as everything spreads out. There's no energy required, that's why we call it passive transport. Simple diffusion, materials move directly through the cell membrane, don't require any kind of transport protein. These are things that are soluble in the lipid bilayer. Facilitated diffusion, materials move through channel proteins or transport proteins, things that can't go through the lipid bilayer alone. But in both cases, they're going to move from high concentration to lower concentration. So here's an example of diffusion in the cell. You have a higher concentration of molecules outside than inside. Solutes are going to have particles moving in both directions, in and out of the cell, but the net movement, more stuff is moving in than moving out, so it goes from higher concentration to lower concentration. And diffusion continues until you reach equilibrium, at which point there's no net change. The amount moving in and the amount moving out are the same. A facilitated diffusion requires some kind of transport protein. In this case, the transport protein is for water, but there's transport proteins for lots of things that can't go through the cell membrane directly. Osmosis is diffusion of water. Water moves from an area of lower solute concentration to an area of higher solute concentration through water channel proteins called aquaporins. So here you can see the process. In this example, you have a selectively permeable barrier that allows water to pass, but not sugar to pass. And you can see a higher concentration of solutes on the right, lower concentration on the left. So the net flow of water is from right to left, and the water rises more so on the right. So when we talk about solute concentration, we use the term tonicity. If the solute concentrations are equal on both sides of a membrane, we say that those solutions are isotonic. If one solution has a higher concentration than the other, we say the one with the higher concentration is hypertonic. If the solute concentration is lower than the other side of the membrane, that's hypotonic. So osmosis moves because of what we call osmotic pressure. The difference in solute concentration creates a net force of water against the membrane. Osmotic pressure would be how much pressure would have to be exerted to stop the flow of water. So here we've got an animal cell and a plant cell in an isotonic solution. Water's moving in and out at the same rate, so there's no net change in the cell. If we place the cell in a hypertonic solution in the animal cell, the water is going to move out. In the plant cell, it's going to lose water primarily from the central vacuole, so it's going to become less rigid, have less sturdiness to it because there's less pressure against the cell wall. We put the cell in a hypotonic solution, water is going to move in, the cell could rupture, or they call that plasmolysis. Water moves in, pushes against the cell membrane in the plant cell. Active transport is similar to diffusion or passive transport, except it requires energy. The form of ATP. And it moves materials against the concentration gradient. It takes them from where they're in lower concentration and moves them to where they're already in higher concentration. So there's two types: molecular transport. Small molecules and ions are carried across the membrane by proteins in the membrane. An example of this is the sodium-potassium pump in cell membranes of neurons. And bulk transport, where larger molecules and solid clumps of material transported by movement of the cell membrane. There's two forms: endocytosis, which is taking materials in by infolding or pockets in the cell membrane. The cell eats something else, is what the term means. And exocytosis, releasing large amounts of material by fusion of the vesicle or vacuole with the cell membrane. Things that have been made in the cell, packaged into vesicles and vacuoles, that then move to the cell membrane and are released from the cell. So here you can see molecular transport, the protein pumps on the left. In the middle, you have endocytosis, where the cell membrane folds in, or in some cases, the cell membrane may actually surround the particle and form it into a vesicle. And finally, on the right, exocytosis, where the vesicle is fusing with the cell membrane, releasing the material outside the cell. Here's an example of a cell engulfing another cell.